let's kick off today's talk with Whitney Stuard from Dr. Danielle Robinson's lab in the CMB program. Whitney is an MD PhD candidate currently doing research in the ophthalmology department on dry eye disease. We asked the participants to give us a one-liner for their introduction, and Whitney told us that she is actively involved in the community service and health advocacy. Her talk is entitled Envisioning a New Role for IGF-BP3 Restoration of Ocular Surface Health.
Today I'm going to tell you about a disease that affects 16 million Americans and in severe cases can cause disability and blindness. Dry eye disease is a chronic and painful disorder that negatively affects quality of life and has no effective treatment or cure. The outermost layer of the eye, the corneal epithelium, is essential to resisting stress from the outside environment with the protection of the overlying tear film. However, this system is not foolproof, and when the health of the tear film is impeded, dry eye disease can strike. Those of us studying dry eye disease are looking at two main facets. The first one is to determine the mechanism by which the health of the tear film is impeded, and the second is to develop therapeutic treatments to allow us to be able to prevent the damage that we see occurring on the ocular surface and to our patients. In dry eye disease, the tear film becomes more concentrated, and this phenomenon is called hyperosmolarity. And what we've seen is that as osmolarity increases, our corneal epithelial cells have negative impacts to their mitochondrial health. In my previous work, I've looked at insulin-like growth factor binding protein 3, or BP3, and found that it is metabolically protective to corneal epithelial cells under stress. Interestingly, when our cells are exposed to increasing levels of hyperosmolar stress, we find that as osmolarity increases, BP3 decreases. Even more interestingly is that in an animal model of dry eye disease, we find that as length of time with the disease increases, BP3 once again decreases in their corneal epithelium. Therefore, if BP3 is affecting mitochondrial metabolism and mitochondrial health in these corneal epithelial cells, would supplementing it back to cells under acute hyperosmolar stress allow those cells to rescue their mitochondria and maintain their cell viability for longer? The answer to this question we now believe is yes. Not only does BP3 allow the cells to maintain near normal levels of mitochondrial metabolism when they're under hyperosmolar stress, but it's also affecting the mitochondrial quality control mechanisms and allowing for increased fusion to create these robust, elongated mitochondria that are better able to withstand this stress. Now, my work is specifically trying to determine what the mechanism behind BP3's role is and how we can best exploit that for therapeutic techniques to help these patients. Because by understanding BP3's role, we will not only better understand dry eye disease, but one day be able to help those 16 million Americans suffering from this disease. Thank you.